Good morning. I know we have a few still stragglers coming in, but we want to go ahead and get started so those of you that need to leave will be able to leave on time. We're so excited about our presentations this morning. If you haven't heard through the last couple of days, there's a lot of research going on PKU, and we're excited to have our panel with us today. But before I introduce the panel, I would like to announce the results of our survey participation. We had over 30 new participants added to the registry yesterday, Eileen has announced, and over 350 new surveys were completed, which is huge for us. So thank you, thank you. And we have our gift certificate for the winner, which may or may not be here. It's Levi Labor, son of Brittany. I don't know if they're here still. Do we see anybody? Yes? Well, we'll find them and give it to them. They won the $100 gift certificate for our registry. So thank you all. That's really exciting. Go home and finish filling out your surveys and look for some new information on our surveys as we send out those emails. So to introduce our panel, who's going to be discussing the emerging PKU therapies, we have Dr. Carrie Harding from Oregon Health and Science University. Carrie, wave. Chris Carpenter from Rubeus. Dr. Nichols. And we have Albert Seymour from Homology. And who am I missing? Eva Brennan. And Yalt Hausman. And they're each going to give their presentation on the emerging therapies and research, and then we'll have time again after each presentation for some questions and answers. So our first one we're going to have speak is Dr. Eva Brennan from Synlogic.
Hi, everyone. It's wonderful to be here. I have a little bit of a croak in my voice. I'm also Irish and have an Irish accent. I think there is some Irish contingent here. Maybe they've already left. So I'm delighted to be here with you and kicking off this really exciting panel with some great companies and great science that's happening in a really incredible and exciting time, I think, for treatment of rare diseases in general and also for PKU. So to tell you a little bit about my company and the approach that we're pursuing, my name is Eva Brennan. I'm the Chief Medical Officer. I've recently stepped up as the Chief Executive Officer of Synlogic. We're a small biotech company that's based in Cambridge, Massachusetts. We're spun out of MIT, and so we're located very closely with the MIT campus, if you're familiar with that area of Massachusetts. And so what we do is we develop probiotic bacteria for specific therapeutic indications. So I'm going to tell you a little bit about bacteria and the microbiome to set the stage for what the specifics of what we're doing is, because I think there are some similarities but also some differences that you'll hear across the various presentations this morning. So just to give you a little bit of background about bacteria, and the microbiome in general. So there are trillions of bacteria that already exist within us. We've known that for some time. But I think what's become more apparent recently is that they're not just passive bystanders, but that they actually play a very important role in health and disease, because there's a two-way communication between what happens in the gut and what happens in the rest of the body. So even when I was training and going through medical school, the GI tract was kind of considered to be a pipe that went from mouth to anus, and its main job was to absorb food substrates from the gut. You know, I think as more information has started to become available, we've really realized that the process is a lot more dynamic, and with two-way communication between what's happening in the gut and all of the bacteria that live there and what's happening in the rest of the body. So now we think about this schematic of, the kind of microbiome and the dynamic interaction as being one that's, two-way. So you have diet and food that provides really important energy and substrates for the body. The diet that you consume also provides important substrates for the bacteria that live within your gut and stimulates them in different ways to create this crosstalk between the body and the microbiome. So this is very dynamic, interactive, two-way process that goes on with the body secreting things that help support the microbiome, like mucus, that the bacteria in the microbiome can live on. And then the bacteria secreting things into the body that can really help support good functioning and good health. So what we do is we build upon this knowledge and all of this history of taking probiotics to support health, but we make them very specific by applying tools of synthetic biology and genetic engineering so that we rearrange the genetics of the bacteria to do specific things within the gut or within the body. And we call these new bacteria synthetic biotics. So we don't take genes randomly. We use genes from other bacteria that exist within the microbiome and put them within a probiotic kind of chassis, as we call it, to create something that's designed very specifically for the disease that we're trying to treat. So this is our pipeline. We have a number of different indications. We've focused a lot on rare metabolic diseases, and PKU is one of the leading diseases that we're pursuing. And the reason for that is summarized on this slide. You'll recognize Christine and her beautiful boys here. So why do we think that PKU is an interesting indication for this technology? Well, the first thing is that diet is a major source of Phe, right? Everybody in this room, I think, appreciates this. And that Phe, we know that Phe is present in the gut even without dietary input. So Phe travels in and out of the gut. Even when you're not consuming Phe in your diet, there's still Phe available within the gut. We know that bacteria can consume phenylalanine, and what's very important is that they consume phenylalanine in a way that's similar to how we as humans do, meaning that they consume it as small di and tripeptides, not as individual amino acids. We know that bacteria can live in the gut, in the GI tract. There's years and years of, millennia of evolution that supports that idea. And the enzymes within the bacteria can evade the immune system because they live within the bacteria. They can also evade digestion, so the enzymes are not broken down like other proteins and foods. And then really importantly, we had some very early feedback from a lot of people probably in this room. As we were thinking about diseases and disease areas, we got a lot of encouragement from patient participation early on when we were still very preclinical that really supported the idea that there was room for further innovation in the treatment of PKU, which for an early stage startup was really important in pushing us forward towards clinical development, because we had good feedback that we were actually addressing potentially an unmet need from patients. So thank you to those who participated and provided that feedback early on. So the goals of our program are first and foremost to normalize blood Phe, but then also to allow patients with PKU to consume more natural dietary phenylalanine. And we're hoping that this will be available in a presentation very similar to a probiotic type of product that's taken every day by mouth. So this is the kind of schematic that demonstrates potentially how this treatment could work. And it's important to say we're still at the phase of development where we don't know whether it works or not, very early on, doing some preclinical and early clinical studies to see if there's a path forward. But this is kind of the theoretical framework that we have demonstrated occurs in a Petri dish and occurs in animal models, and we're now evaluating in humans. So we have a bacterial species. It's based on a probiotic supplement that's available in other parts of the world. So we've kind of have some human experience from that probiotic. It's available in Germany and Canada and other places under the trade name of Mutaflor. People take it as it's thought to help with gut regularity and also help maintain remission in patients with inflammatory bowel disease in those areas where it's available. And then we engineer it by taking genes from another bacterial species that lives within the human microbiome, and we put in a PAL enzyme and another enzyme called L-amino acid deaminase. And so the idea is that the bacteria, as they're transiting through the gut, will consume phenylalanine continuously and produce the products of those enzymes, tranexamic acid, which is conformed to HA, called hippuric acid. And the important thing about this engineering, as you'll see later, is it allows us to evaluate the activity of the bacteria in some of the preclinical species, because we can measure the product of those pathways and evaluate, excuse me, that the bacterial circuits are being switched on within the human gut system. So as everyone does in PKU drug development, we've evaluated the bacteria in a number of different animal models. Some of those studies are designed to evaluate efficacy, and some of those studies are designed to evaluate safety before we move forward into the clinic. So the animal model that we use quite extensively, I'm sure is very familiar to most people in this room, is the Inu2 mouse model. And that animal model has a mutation that's similar to the human form of PKU. And so we do a construct where we give them, they're maintained on a low-Phe diet, and then we give them systemic phenylalanine, and we measure how much of the injection Phe can be taken up by the bacteria in the gut. And we're demonstrating that that when we administer our bacteria, that you're able to see a reduction in this blood Phe levels in that animal model. So it's an acute model that we use very frequently to evaluate kind of how active we're being in the GI tract. We've also done a lot of studies in healthy non-human primates. So these are monkeys that have normal PAH enzyme activity, but because we're able to measure some of these, what we call pharmacodynamic biomarkers, that's the TCA and the HA, we can evaluate what the activity of the bacteria are within the gut system. And monkeys are much more similar to humans in terms of their GI tract and their transit time. So it's a good animal model for us to look at that gets us closer to what the human would look like. The important thing, I think, about this one for us, and that's probably a little unique compared to some of the other programs, is that it's one of the challenges is that it's hard to sample the gut continuously. So you can measure what goes in the top end and you can measure what comes out the bottom end, but in the middle, it's kind of difficult to sample continuously. So it's very hard for us to get an idea of what the bacteria are doing once they get inside, past the stomach and down into the intestine. But the nice thing here is that we actually have this activity biomarker that we can measure that tells us that the bacteria are getting into the small intestine, that this circuit that we've designed is switching on, and that it's doing its thing. And we can also look at the association between how much bacteria we're giving and how much of the biomarker we're seeing in the blood and the urine. So it gave us kind of a nice preclinical model system where we could really look at the relationship between the dose of the bacteria and how much activity we were seeing, which was very important because of this kind of inability to sample continuously from the human gut. So this was all very encouraging. We had some discussions with the FDA. We submitted, the first step in clinical development is to submit what's called an investigational new drug application, which we did earlier this year, and we're currently in early phase clinical testing. We're planning to do some studies in healthy volunteers to really understand safety, tolerability, look at the relationship between dose and the biomarker that we identified preclinically. And then once we think that we have a path forward, we will move forward into patients with PKU, probably in over the summer and the second half of the year. So we have some information here about where to find more information about the study, and we have a number of sites that are getting up and running right now where patients with PKU can participate, and the link to clinicaltrials.gov for anybody who'd like more information. So that's it for my remarks. I'm happy to take questions now or later, whatever works for the moderator.
Good morning. In your non-human primate test, did you encounter problems with infection? I understand that's often a side effect of probiotics. Did you have any problem with that?
Yeah, so I think I'll just repeat the question to make sure I heard you correctly. So the question was whether we've seen any problems with infection in the monkey studies. So I showed you a snippet of the preclinical work that we've done. We've done some toxicology studies as well. We've looked at mouse and non-human primates. I've never seen any issue with infection. And we also have done a lot of review of the parent probiotic strain to the extent that there's information available looking at big studies, and there's never been a case of infection with the parent strain either. But we're very early in development. Obviously, that's one of the things we're looking out for as we go forward. But so far, based on the evidence with the parent and the evidence that we have in the preclinical species, we've seen nothing with infection. And an important component of all of our studies is that we make sure that the bacteria that we're putting into the clinical trials are sensitive to a full panel of antibiotics. So that's something that the FDA requests us to do and that we do before we move forward. So hopefully that answers your question. Great.
All right, thank you. And next we have Dr. Chris Carpenter from Rubeus.
I'd like to thank Michelle and Christine and the Alliance for the opportunity to tell you today what Rubeus is doing. And tell you too personally, it's been a real pleasure to be here, to see how families and patients and academics and industry are all able to work together really is, I find, inspiring and refreshing. And it's quite different from any other meeting I've ever been to. So thank you for that. Rubeus started about 4 years ago, and the whole idea is that red blood cells can be used as therapeutics, not in the sense of transfusion, but at much lower doses. And I'm going to tell you first a little bit about red blood cells, then what our platform looks like, what we're aspiring to do using red blood cells, and then focus on what we're doing in phenylketonuria, which we hope will be our first clinical program starting early next year. I'm a hematologist by training, and I have to tell you, when I actually took a step back and looked at what red cells do from the Rubeus perspective, I was still a bit astounded that red cells have one purpose in the body, and that's to transfer oxygen from the lungs to the tissues and the rest of the body. They do it through hemoglobin. But I didn't realize they were 80% of the cells in all of our body are red cells, and we're making over 2.5 million of them every second. So they really are a key part of our entire body, the way they function. What we're hoping to do is just co-opt a bit of what red cells do normally, transporting oxygen, getting it around the entire body, and make them into drugs that can help people with rare diseases through enzyme replacements, with cancer, and ultimately, we hope, patients with autoimmune disease as well. There are a couple of key features that red cells have. As I said, they're the most common cell in our body. They last a long time. When they're made normally in the body, they last about 4 months. And they all tend to die at the same time. They express normally what are called a protein called CD47, you may have heard about in some of the other talks, which is what's called a don't eat me signal. There are cells in the body called phagocytes or macrophages that will recognize cells that are becoming sick, need to be replaced, and will eat them basically. So red cells express this CD47 protein to keep them surviving, but at some point they stop expressing it as much, there's some changes to the cell surface and they're destroyed, and this all happens about at once. So there's very predictable half-life of red cells. There's also a very predictable what we call biodistribution, meaning we know where they are. They're red blood cells, they're in the vasculature, they're in the arteries, the veins, and in the spleen, and that's about the only places they go in the normal body. One key feature of red blood cells, especially for what we're doing, so we're genetically engineering red blood cells to make them express proteins they normally wouldn't express. But to do that requires that they have a nucleus, which they do early on, but as they mature and become a true red blood cell, they lose that nucleus. And so as they lose that nucleus, they lose the DNA and anything that we've introduced. So it has the potential to reduce any risk that might be associated with genetically engineered cells. Before I tell you how we're making red cells, which is completely outside the body, I wanted to tell you first how the body normally makes it so you can appreciate the contrast in what we're doing. So all of our blood products in our body are made starting from a single class of cells called a hematopoietic stem cell or progenitor cell. So that's a cell that can give rise to all of our white blood cells that fight infection, our red cells that I said that carry oxygen, and platelets that help the blood to clot. This happens in the bone marrow, so the hollow spaces in our bones, and you can see on the upper right image is a photomicrograph of what bone marrow looks like. So those pink strands are little pieces of bone that sit inside the bone marrow. The white spaces are fat. There's actually a lot of fat in our bone marrow. And then the pink and bluish areas are where the blood is being made. Red cells start from that hematopoietic stem cell and then differentiate all the way through various stages and end up, as I said, at the very end, spitting out their nucleus and end up as not quite a bag, it's much more sophisticated than that, but primarily a cell without anything other than hemoglobin in it to transport oxygen around. When they're developing in the bone marrow, they develop in what are called these erythroid islands. That's the second image you can see on the right. There's what's called a macrophage, one of these sometimes phagocytic cells that sits in the middle, provides the right environment for the red cells to develop. They can stick to it, it provides the growth factors they need, and they develop all around this macrophage until they lose their nucleus and then leave the bone marrow. And then in the lower right, you can see what normal red blood cells look like on a blood smear. They're round and they're red from the hemoglobin. And that's very much like what our red cells look like. And I'll show you a photomicrograph of ours in a few slides. So at Rubeus, we start with the same cell. So this class of cells, these hematopoietic stem cells or progenitor cells, are identified based on some proteins expressed on their cell surface. One of these is called CD34. So it's a protein that's on the cell surface and so can be used to purify these cells. There are antibodies that exist that recognize CD34 and can pull them out of the mix of every other cell in the body. And that's how we get cells to start with. It's the same process that's used for people who are going to donate blood or cells for bone marrow transplant. That patients who need a bone marrow transplant need these same cells to reconstitute their entire blood system. And so we purify them the same way, but rather than them going to bone marrow transplant, they come to us, and then we freeze them down in aliquots, and if we want to make red cells, we thaw one of those aliquots, and then go through a three-step process to make red blood cells outside the body in big vats. And I'll show you a photo of one of those in a minute. The first part of the process is we expand these cells. So there's still these hematopoietic stem cells or progenitor cells that can give rise to any of your blood cells. So we expand those. Then at the end of the expansion phase is where we do the genetic engineering. So we use a lentivirus that's expressing a gene of interest and use that to transduce the cells. And in the case of phenylketonuria, we're using the Palynziq and introducing that through a lentivirus. And then we go through the rest of the process. We call it a differentiation phase and finally a maturation phase. So we change the environment the cells are in to make them become red cells. And then at the very end, like normal red cells, they lose their nucleus. And then we harvest them, we filter them, we formulate them, and then they're ready to give to people in very small volumes. We're thinking, depending on what the indication is, probably around 25 milliliters, which would be a quarter to a half a cup at most. On the left is a photo of actually us making red blood cells. This is one of the bioreactors we're using. We're making them in bigger and bigger bioreactors now. We're making them in 50 and up to, we're moving now to 200-liter bioreactors to supply what we think is going to be necessary for a clinical study. And on the right are the photographs of our cells. So if you remember back to what they look like normal human blood cells, these look exactly the same on the upper right. And then on the lower right is what's called a scanning electron micrograph. So it's a way to look in more detail at a bigger image of a red cell. And this looks exactly like a normal red cell. It's round. All normal red cells have what's called a biconcave shape, almost a donut, but they have a little bit of membrane in the middle. So they look exactly like we would hope they look. And these are expressing the Palynziq. There are a couple of twists on doing this that are really important to understand. And You probably know, if anybody's ever gotten a blood transfusion or know somebody who does or know about blood transfusions, that it's really important to give somebody the right type of blood. And there are two factors that are important in typing blood, what are called AB antigens and what are called RH antigens. So you can see on this slide that there is illustrated people can have either the A antigen or the B antigen or both or neither. So they're called A, AB, or O if they don't have either of those two antigens. And combined with that is RH, so the RH antigen. So you can be A positive, meaning you have the RH antigen, AB positive, B positive, or any of those negative, or you can be O positive or O negative. So O negative, people who are O negative are called universal donors because the blood they donate can be given to anybody because they don't have any of the antigens, so nobody will develop an immune response to those blood cells. And those are our donors. We want to develop a product that can be stored and then given off the shelf to anybody. And so it's important to start with donors who are universal donors. So the products that we make won't express any of the antigens that people might react to. About 6 to 7% of the population fall into this group of being O negative and RH negative. Just before I tell you a bit more about PKU, I'll tell you a bit about what we're thinking the potential is for the approach that Rubeus is taking in using red blood cells as therapeutics. As I said, they're going to be ready to use, so we're using universal donors, so we don't have to type the recipient's blood to what we have on the shelf. We know it will be ready to go right away. We think there will be lots of applications. So our first, as I said, is going to be phenylketonuria, but we think there are other rare diseases and enzyme replacements that are also amenable to this approach. We think that patients who have cancer may also benefit from the things that we can do with red blood cells, as well as patients with autoimmune disease. So in the future, we hope to be exploring these areas as well. I mentioned earlier that we know where these cells go, and that's a real advantage to know what their biodistribution is, and that they stay within the blood system. They stay within the arteries, veins, and the spleen. We think that they're going to have a very long half-life, which in most cases is going to be a real advantage, meaning that we can give small doses and infrequent doses. As I've said, that to date, everything about these red cells looks like a normal red blood cell. So we're hopeful that when we give them to patients, they'll have the same lifespan, close to 4 months, as normal human red blood cells do. And finally, we think this is really going to be, that there's a long history of blood transfusions going back decades and decades. So many of the safety issues have been identified and worked out, and so we can follow that track that's been set out about how to work safely with blood products. And then, too, we think this is going to be quite scalable. As I said, we've been moving from smaller vats to bigger vats, and even bigger ones are on the horizon as we make the product that we're going to use for a clinical study. So we think it's going to be easily scalable to provide the product that we're going to need. We're calling the product for PKU RTX-134. PKU was discovered in 1934. We couldn't figure out any better way to come up with a number for our first product, and so that's what we've decided on. This illustrates basically what I've told you to date, that we're making a red blood cell that's expressing the Palynzyme. I'm sure you're quite familiar with how it works. It takes the amino group off phenylalanine and turns it into tranexamic acid and ammonia, and then the tranexamic acid is metabolized to hippuric and benzoic acid and primarily goes out in the urine. And based on the work that BioMarin's done, all of this seems to be quite well tolerated by patients, so we think in this setting it will be as well. We think there are some potential advantages to the approach that we're taking, that the red cells, we think, will last a long time. Because they're inside the red cell, they'll be shielded from the immune system. And even if antibodies do develop to PAL, then it shouldn't affect the activity of the protein within the red cell because the antibodies can't get inside there. So we think that it can provide an advantage in avoiding immunogenicity to PAL that could impact its activity. As I said, we think it's going to be dosed infrequently in quite small doses. We don't think of this at all like a transfusion, just an infusion. It will need to be given by vein, but again, we think quite infrequently. We've talked already with the FDA, had preliminary discussions, and they were quite encouraging. We plan to file what's called an investigational new drug application, or ask them for permission to begin a clinical trial early next year, and hope to begin the trial shortly after that. Our trial is going to be a fairly typical phase 1 trial, where our goals are to be sure that the product is, first of all, safe, and then to begin to find out what the correct dose is and find out whether it's working well, as we would hope it would. This is a bit of preclinical data. We don't have a lot, and the reason is that giving human red blood cells to mice is very much like giving mismatched human blood from one donor to a recipient. The mice destroy them right away. So it's very hard for us to get any valid preclinical data in a mouse model. But what we've done, I'll show you on the next couple of slides, is first predict what we need for a dose. And this is based on modeling we've done, based on work that BioMarin has published, some preclinical work that we've done, and preclinical work that others have done. And on the slide on the right, you can see we know how much activity we need per red cell in order to reach what we think is the right therapeutic dose. And right now, we're able to exceed that by quite a bit. So we think we've got able to produce PAL in the red cells at a level we need to have the therapeutic impact we hope to have. On the left here, we've looked just outside, not in mice or people yet, but just in a test tube to look at the activity of PAL expressed in the red cells. And as you can see, it quite well degrades phenylalanine, and there's a dose-response relationship exactly as you'd expect. The data on the right are a bit complicated, and they're about as good as we can do in finding out how the red cells are going to do in mice. I'll walk you through it. So what we've done is taken the lentivirus that expresses the PAL protein and transduced it into the early hematopoietic cells, and then given those back to mice, replaced their bone marrow. So the mice are now growing blood cells, all of them, that have the PAL enzyme expressed in them. We've then taken the red cells from those mice, given them to other mice, and then looked to see how long they survive. A bit of a complicated experiment, but what it's allowed us to conclude is that the red cells survive as long as normal red cells do in mice, which is 40 or 50 days. So again, very encouraging that the cells we make, human cells we make, will have a long survival in humans, approaching that which the normal cells do. That's my last slide. This is the group that's made it all possible. Not all of them, we wouldn't all fit in the image. We're up to over 85 employees now. We're in Cambridge, Massachusetts, continuing to grow. This is the leadership team at Rubeus. And really thank you for your attention, and happy to take any questions.
Do you intend on using hematopoietic stem cells from O-negative donors and then growing them up and transfecting them in culture and then using those for the transfusions, or?
That's basically the process, yes, we do. We get the CD34 positive cells from O-negative donors. We get a lot, so we freeze them down in aliquots. But then as we start the process, yes, we just thaw one of those aliquots. After a few days of expanding them, when they've reached their maximal expansion, then we do the transduction. And then we continue the differentiation on into a red cell after that. Yeah. Either place. I'm happy. Yeah, go ahead. Yeah, it looks like we're ready over here.
So is there any chance of acidification of the blood if you're creating all of this cinnamic acid in the bloodstream?
I'm sorry, any chance of creating
acidification of the blood?
Acidification.
Acidification of the blood or of the body system?
No, we're formulating in a formula that won't do that. It's the same pH as normal blood, and we're giving very small volumes relative to the normal blood volume. Even at the highest dose we anticipate ever giving, the red cells we give will be less than 1% of the total red blood cells in the body.
Hi. So how do you think about a batch of drug in this context? Is it pooled donors that you then transfect with the lentivirus to create a batch, or is it single donors that you expand, to make a single dose of the...
Single donors. And I don't know whether you're getting at this or not, but the issue is that although the major antigens that determine blood compatibility I told you about are A, B, and the Rh antigens, there are some minor antigens, and a few of these can cause reactivity in patients. And so to, first of all, be able to track that down if it ever happens, we're using single donors. And we think it's going to be a better way to provide the product is always coming from a single donor. Now, that would be for a single dose. For different doses could come from different donors. And we are planning to do what's called cross-matching. So we will check the patient's serum, which has antibodies in it, for any cross-reactivity with our cells before we give them, to be sure it's safe to do so. It's a standard blood transfusion procedure that we're adopting.
Right. So over time, a patient would be exposed to different donors' cells in that case.
I'm sorry, I heard the first part, over time about exposure to different donors, but I couldn't.
Yeah, over time, they would, the first dose might be from one donor, the second dose might be from a second donor, et cetera. So you'd be exposed to multiple donors over time.
That's correct, yes, yeah.
So when you say small dose, I'm just curious, like what volume are we talking about? Like what's considered small?
Let me make sure I heard you correctly. What's considered a small dose?
Yes, like the volume. Like,
Yeah, we're thinking, so 25 mLs, which would be about a quarter of a cup to half a cup or so,
Okay.
and we're hoping that would be somewhere between 1 and 4 months.
Okay, perfect.
Hello, I have a question. Close,
Thank
sorry.
you.
So if a patient will be exposed to multiple donors, eventually he will be sensitized to develop antibodies to different donors. So we still will expect at certain point he will develop kind of rejection or reaction and expected to be medicated with prednisone or maybe immunosuppressed drugs. How do you see it over the path of years of therapy?
So we don't know whether antibodies will develop or not because our product is encapsulated within the red cell. We certainly will be monitoring for antibodies during our clinical studies. If they do develop, we don't anticipate they're going to have any negative impact, either from a safety or from an efficacy standpoint, because the enzyme in our case is inside the red cell. It's got that membrane, and antibodies can't get inside that membrane. So we don't think that there's going to be any adverse impact, even if antibodies do develop.
I'm sorry, just one last question. So regarding the donors, for you to scale this, if this works as we hope it does, you're going to need more donors. If people stop donating blood, does the stream stop? I'm thinking about the source. Do you need more and more donors as you?
We do, but we've calculated, as I said, about 6.5% of the population are O-negative donors. We've calculated how many we need over the coming years, and it's easily achievable for us.
Any other questions?
Thank you again.
We next have Dr. Carrie Harding to talk about Palynziq.
So good morning. I'm going to magically reappear this morning. So it's a great pleasure and privilege for me to be able to present to you on behalf of the really large team of people that participated in the Palynziq studies, to present to you on Palynziq, which is a novel enzyme substitution therapy that's been recently approved for use in adults with PKU. So I had the honor to serve as the chair of the steering committee that advised BioMarin on the conduct of the clinical trial. So I have to disclose that I was paid as a consultant to the team, but I don't have any rights to the drug and I don't own any BioMarin stock. So we have accumulated a vast amount of data, both on the safety and the efficacy of Palynziq, and treating over 350 patients now with Palynziq over 10 years. And we only have time really to highlight just a little bit of the data. So if you're an adult that's considering using Palynziq, I really encourage you to talk to your PKU provider, and they will have the availability of seeing a lot more data that they can discuss with you, both about the safety and the efficacy. And I'm only really going to cover what's available in the prescribing information, and there's a lot more published information out there. The thing I want to really drive home, safety is our central concern. And the most important information that you need to know is that Palynziq can cause severe allergic reactions that can be life-threatening. And consequently, BioMarin has developed a risk evaluation and management strategy for the patients and their providers. And this has been developed to help prevent and manage any severe reactions. So let's discuss how Palynziq works, and then we'll talk about the efficacy that we saw in the clinical studies, talk about the safety profile and how this would be administered at home. So as many of you know, the buildup of Phe can both damage the brain but also cause significant adverse effects, such as things like forgetfulness, trouble focusing, difficulty carrying out complex tasks, symptoms like anxiety and depression. And Palynziq really works in a completely novel way to reduce phenylalanine in the blood and ultimately the brain. And Palynziq treatment is not dependent upon reducing dietary Phe intake. Palynziq is a daily at-home injectable treatment. The first injection of Palynziq is given in the office of your healthcare provider, and thereafter, once you've mastered doing injections, you would do this at home. Palynziq's been proven in the clinical trials to significantly lower blood Phe, and in the clinical trials, in those patients that were taking the 20 milligram per day dose, we saw an on average 62% reduction in the blood Phe, and I'll discuss more about the effectiveness of Palynziq later. Most patients taking Palynziq experience side effects. We've learned a lot about dealing with those side effects, and you'll definitely work with the healthcare provider to help manage any of those if they were to happen. So how does Palynziq work? So it works as a substitute for the PAH enzyme to lower the Phe levels. It's been shown to work in all types of patients with PKU, so it doesn't really matter what your genotype is. It doesn't matter how much PAH enzyme activity you have. This enzyme works independently of what your PAH enzyme activity is. Palynziq breaks down Phe, as you've already heard from the other speakers, into tranexamic acid and ammonia. Those metabolites are then handled through your liver and ultimately excreted in urine. And by keeping the Phe levels down, there's less risk, obviously, of this toxic buildup of the Phe and keeping the levels low throughout your body. So as I mentioned earlier, the patients in the phase 3 trial who got the 20 milligram per kilo dose experienced, on average, a decrease of a 62% reduction in blood Phe. So before treatment, before they went into the trial or started, the average blood Phe was 1,450 micromolar, or 24 milligram per deciliter. By the end of the study, that is the pivotal trial where we did the placebo control arm, those patients had dropped their blood Phe down to 553 micromolar, or 9 milligram per deciliter. So that's a reduction of 897 micromole, or 62%. Sorry, I meant to go back. And I want to point out that virtually all of these patients were not restricting their dietary phenylalanine during this period of time. So you need to be aware that it can take time to find the dose that works best for any individual. We worked hard in analyzing all of the data to try to figure out from something early on that would predict who would have reactions, and what dose someone would need, and we were not really able to ever find a marker that would tell us that. So basically, you just have to start on the program and slowly work your way up in dosing, and the timing of all of this is managed by your reactions and the Phe levels to ultimately find the best dose that would work. And it can take longer than 8 months, and in some cases up to even a year before you and your healthcare provider find the right dose. And it really all depends on how you would individually respond to therapy. Everyone gets antibodies against the drug because it's a foreign protein. But everybody responds differently to those antibodies. Your body may see this as a very foreign thing, and that can cause side effects, including anaphylaxis in a small number of patients, and this can be a serious problem. Your healthcare provider obviously would discuss the risks with you before you would start Palynziq. If these happen, you obviously need to stop injecting the Palynziq and get emergency medical care right away. If you had any of a list of symptoms of a severe allergic reaction, I'm not going to go through the specific list of symptoms. The most common thing that we saw were injection site reactions, where you get some redness and itching around the injection site, and beyond that, you tend to advance towards a more generalized skin reaction. And then the next most common thing, as I'll get into in a minute, is more severe joint pain. Those are not the severe anaphylactic reactions that needed to be more significant, significant emergency treatment. Next, we'll discuss, though, the Palynziq REMS program. So Palynziq's only available through a restricted program that's called the Palynziq Risk Evaluation and Mitigation Strategy, or REMS program. The purpose of the program is to ensure that the patient understands the risks and benefits of treatment with Palynziq. You would talk to your healthcare provider, or their BioMarin has account managers that would give information about the REMS program and how to enroll. You can also go online. There's a website, palynziqrems.com, that gives more information about this. So this is more information about the side effects. They can occur at any time during treatment. In the clinical trials, nearly everybody taking Palynziq experienced some side effect at some time. Most of these side effects can be effectively managed. The management includes slowing down the rate at which we titrate up on the dose. It can include pre-medicating with antihistamines and with anti-inflammatory medications. Most of the side effects are mild to moderate. They generally decrease over time. Most of the side effects occur early in the treatment, which is during the titration phase, and they lessen with time as the antibody response matures. As I mentioned before, some of the patients got joint pain during this early phase, and although a lot of patients experienced some joint pain, most of this was less than 2 weeks. Occasional patient had this for more than 2 weeks, and again, we've learned some strategies for trying to deal with this. This is kind of a common sample treatment plan, but I want to stress that it ends up being very individual, and we haven't been able to predict, again, up front, what's going to work for any individual patient. So this is just a guide, and you have to work with your healthcare provider to really figure out what works. There are 3 phases of treatment. There's induction, titration, and then basically the ongoing maintenance therapy. And the goal of the treatment schedule is to gradually increase the dose and frequency until you find the right dose that works. So you start usually with 2.5 milligrams once a week. That usually is for 4 weeks. And then you go to twice weekly, and then gradually increase the dose and the frequency of the dose up to 10 milligrams once weekly at first, and then twice weekly, and then 4 times weekly, and ultimately daily. And then you try to push up from there up to 20 milligrams a day. Again, watching Phe levels and changing the frequency of both the dose and how often you give it, depending upon whether or not you're having any significant reactions. Ultimately, you and your healthcare provider will decide on the specific lowest effective dose, and we want to stay at 20 milligrams for at least 24 weeks to see if that's effective. If your Phe levels haven't dropped by that time, then we consider going up to 40 milligrams to see if that will work to lower the blood Phe if you've not had a response to 20 milligrams. Again, this particular program that's shown here is just a guide, and you would develop an individualized treatment program with your healthcare provider. It's also important during all of this to try not to make many changes in your dietary therapy until you've been instructed by your healthcare team to do so. So the buildup of Phe, as I mentioned before, can both damage the brain and cause some of these side effects that we've discussed. This is a novel approach to treating PKU. It's an at-home injectable therapy. Definitely has been proven to lower blood Phe while consuming normal amounts of protein, which is what I want to stress. And as in many of our patients managed, we've been able to manage these common side effects and ultimately been able to get through this during the titration phase and get them to where they have an effective dose and have been able to lower their blood phenylalanine levels. This is my final slide. As you heard last night, it really took a total huge village to bring Palynziq through preclinical development, the clinical trials, FDA approval, and now to market. And I want to give a little bit more history. The idea to use PAL as a treatment actually has been around since the 60s. And there was an attempt to use a crude form of PAL as an oral drug actually in the 70s. There were some patients given this as an oral preparation early on, and it didn't really work very well because it got digested by the stomach juices way back when. And it wasn't until Christina Sarkissian and Dr. Charles Scriver's lab at McGill University in Canada had the insight to try bypassing the gut and injecting it into the mouse that we saw a therapeutic effect from PAL. And then it took the efforts of Ray Stevens and people working in his lab, and Christina Still and scientists at BioMarin who did a lot of work to further optimize the enzyme and altering it to come up with a formulation that would really work as a clinical drug, and that was efforts that were largely funded by donors like yourselves. And then ultimately BioMarin taking it on and doing the clinical trial. We did multiple phase 1, 2, and 3 clinical trials that ultimately took 10 years, took 33 clinical trial sites and 355 patients. It's to date the largest trial that's ever been done in any rare metabolic disorder. So I think everybody needs to be extremely proud of that. And it's impossible for us to really individually thank everybody involved. It's literally hundreds of people. But I personally want to acknowledge the adults with PKU that participated in the trial. I personally have really learned so much from them about the unmet treatment need and how important novel therapies are to the adults, and it continuously encourages me every day to keep going back to the lab and trying to find better paths forward. And I really tremendously admire the courage of the people that stepped up, and they had no idea what was going to happen the first time they took the shot of this completely foreign protein, and they're the ones that really took the risks, not me given the shot. And I really applaud them for participating and stepping up and do this. And finally, I really thank you for your attention. So thanks. Questions?
Thank you very much. Among the patients that participated in the trials, did you have patients that had a lower Phe level than the one that you had on the max, let's say around 5 milligram per deciliter? And if yes, did you observe any Phe reduction for them too, or only for the patient that started with a much higher blood Phe level concentration?
Not sure I quite understand. So are you basically asking, did we ultimately get Phe levels that were lower than 500?
No. In one of your slides, you did show that the average Phe level of the patient was around 1,450, if I don't remember well, and that you observe a 62% reduction. My question is, among these patients, I believe that this 1,450 is an average. Among these patients, did you have patients that had to start with a more reasonable blood Phe concentration?
They had to be over 600 to get into the trial.
Okay, so among the ones, they were around 600. What percentage reduction did you observe?
So there were a few that were like barely over 600. Most of them were much higher than that. So most of these patients were consuming, I mean, we only had 15% of patients that were, this is the phase 3 trial. So the final phase 3 trial had 286 patients. And of those, there were only 15% that were actually on what we called a restricted diet, meaning that they were taking more than 75% of their diet as medical food. And even with that, the majority of them had blood fees that were much higher than 600. So although they had to be over 600, most of them were significantly higher than that.
So the answer is no. There was like not really
Basically,
patients. Thank
no.
you.
So I think, so the 1450 was the group of patients that, so the way the trial was designed was they got randomized at the beginning to either 20 or 40. And ultimately, they were able to adjust up or down depending upon what their blood phe levels were. But because of the way the trial was set up, they got randomized. So the data I showed you were the people that were on 20, not the people that were on 40. Taking all of the patients together, I think the starting blood fees was like 1,200 on average. But the range, it was all pretty tightly clustered around 1,200 or so. So probably, but again, they started over. They all had to be over 600 micromolar.
Thank you.
So and the average phenylalanine intake at the beginning was something on the order of like 7,000 milligrams of phe. So that clearly was way more than most of them should be taking in terms of what they should have been in terms of dietary therapy.
I noticed that some people had no response at the 20 milligrams. Did you have anyone that had no response at 40?
Yes.
Do you have a theory of why or a percentage?
Okay, so I'm in verge of getting myself in trouble here. And I'll tell you why. The label, they've licensed 20 milligram and 40 milligram doses. We studied higher doses. The company is continuing to study higher doses. And the hope is that there will be the possibility in the future of being able to use higher doses. It's probably related to antibodies. Some of the antibodies do neutralize the drug. And again, we have not been able to predict who will need what dose. We just have to keep trying. I would just refer you to talk to, if you're interested, a healthcare provider or one of the medical representatives from BioMarin about specifics in terms of exact numbers of how many responded to what dose.
So a lot of people have been saying that they can pretty much eat whatever they want. So since you have to be above a 10 level, do you recommend people who originally have a lower level to try to bring it up to be able to take it?
I don't recommend anything. I think that's a discussion you and your healthcare provider need to have.
Dr. Harding, my question is, you mentioned that hypophy was a common side effect for patients when they were starting this therapy. I'm wondering what
Did you say hypophy?
I'm wondering what
I didn't mention it, but we will acknowledge that that has occurred.
Okay, so I'm wondering what are some of the short and maybe long-term risks for that? And is that any worse or is it less worse than potentially patients with hyperphy?
I do not think that we want to promote having hypophenylalaninemia, but and part of this was an artifact of the way the study was done. So because we had fixed dosing for a period of time, there were patients that had quite low blood Phe levels. Once we were able to adjust the dosing downward for some of the patients that were on more, we're able to let those levels come back up some, or the other alternative was to increase their protein intake. But honestly, we did not, and we worked pretty hard to see if there were any major adverse effects from that, and weren't able to convincingly really find anything wrong with being low. This is different though than, because these patients were getting lots of protein. This is different than having low blood Phe from being overly restricted on diet, because when that happens on diet, that's really protein malnutrition. It's not just too much low Phe, it's lots of low Phe or lots of low from lots of things, and so it's a different situation. I'm not advocating that that's what we should shoot for, not in any stretch, but and we should adjust the doses to try to not have that happen. But it was not a major, we did not see any major problems from it during the trial.
We can take one more question.
Dr. Harding, the trials, or the pivotal trial was based upon you had to be over 600 micromoles to be an eligible volunteer. But the FDA label, just want to double check and clarify something. Is there any restriction that you have to be over 600 micromoles in plasma Phe in order to start?
The current indication is over 600.
Okay, and indulge me, but not a scientific question, but it went from PEG-PAL to PEG-Valase to Palynziq. Why?
I'm sorry, I didn't understand the last part.
Sorry, the name of the product has changed over the development stage from PEG-PAL to PEG-Valase to Palynziq. Any reason why? Other than marketing?
We probably ought to avoid using PEG-PAL, although I fall into that all the time, because there are different PEG-PALs actually. So the original species that Christina used is a different PAL from a different bacterium than is ultimately used for the clinical product. And they pegylated different forms and tried different forms. So the form that ended up being clinically used is from Anabina variabilis. So that became the pegylated form of PAL from Anabina variabilis became PEG-Valase, which is the generic name. So PEG-Valase specifically refers to that species of PAL, and then the brand name of it is Palynziq.
We have a lot of interest, so we're going to take one more back here.
Okay, yeah, Dr. Harding, you may have covered this earlier, but you mentioned that a number of the participants in the trial experienced side effects early on and that they subsided over time. Can you speak a little bit about, what that looks like after folks have been participating, or taking the Palynziq for some extended period? Do the side effects generally tend to go away?
Yeah, they get significantly better. They can happen later. They tend to be, again, early on when we're changing doses. So the mechanism is pretty clearly antibody related. So we just didn't have time to show all these slides and data, but at the beginning, the antibodies show up exactly on day 10 after they get their first injection. And the first antibodies that show up are actually against the PEG part of the molecule. And it's the PEG, and it's the anti-PEG antibody in one particular form of it that seems to correlate most with the reactions. And it's related to if you put a lot of drug in at the same time that there's antibodies, the drug and the antibody bind, and they cause a specific cascade of inflammatory chemicals in your body that cause skin rash, cause joint pain. And so in my experience, and different PIs did different things, but my experience was if you backed off on the dose for a while until those antibodies kind of matured over time and the levels changed, most of those reactions kind of were minimized for a period of time. But you might have to wait 6 or 8 weeks for the antibodies. They don't go away, but they change. So then if you waited a bit and then started pushing the dose up, you might still get some, but they were less severe, and ultimately the incidence of these problems got better. We still had patients later that would have an occasional reaction, but less severe. But the incidence is very clearly much higher in the first probably 2 to 3 months and much less later. And it has to do with the antibody changing over time.
All right. Thank you. And next we have Dr. Hausman.
Thank you, and good morning, everybody. I want to thank the organizers for including Codexis in the program here today. This is my third time at the NPKUA conference, and the first one was in 2012. We had just started our program. We were with 2 people, and I had a dream at that point that maybe one day I would be able to stand up here and speak about our program. And of course, that would require that the program was going well. And we are excited about where we are today, and I'm excited to share our program with you here. I work at a company called Codexis. We're in the San Francisco Bay Area. Codexis has been around for 20 years, 16 years as Codexis, and 4 years before that as Maxigen, its parent company. I've been there all that time. I'm an enzyme engineer. I'll talk a little bit about what that is. In full disclosure, Codexis is a public company, and I'm a shareholder in the company. So, of course, this audience does not need an introduction to enzymes. But I want to start building a link here between enzymes and enzyme engineering. So, of course, you all know that the enzyme phenylalanine hydroxylase, when that has certain mutations, it becomes inactive or perhaps just slightly deactivated, which leads to the accumulation of phenylalanine, and that causes the symptoms of the disease. And a different enzyme that we have learned about quite a bit today and in the previous days and also in previous conferences, we have heard about the PAL enzyme. I have PACPAL on here, but I guess I should have said PACValase, as an example of an enzyme that can take phenylalanine and convert it to something different. So rather than bringing the active, the PAH enzyme back into the body, we look to bring a PAL enzyme that converts phenylalanine to cinnamic acid. And again, in this presentation as well, the relevance of cinnamic acid will become clear, as it is a very good mechanistic marker for the enzyme. Now, as you have learned in the session on Friday, a great overview by Betsy Bogart on Phe metabolism and what is happening with phenylalanine in the body and how we can interfere with phenylalanine metabolism. There's really 2 ways of doing that. The first way is making sure that there's just less phenylalanine coming into the body. The second way is making sure that any excess of phenylalanine in the body is being removed in a different compartment, such as the blood, such as in the liver, through different types of technologies. Where these 2 meet, the removal of Phe or the prevention of Phe in uptake versus the removal of Phe in the body, is in the intestinal organ. And in the intestines, so when food comes into, when we eat, the food and the protein enters the stomach, it's a pretty acidic environment. It's pretty harsh for protein. They start to unfold and become deactivated. They then go into the food. That food mixture then goes into the upper intestines, first in the duodenum, and there's proteases secreted in the duodenum, and these proteases will secrete protein. And that is, of course, the food protein, but it is also any enzyme, a therapeutic enzyme that you may want to introduce. The proteases convert the protein to amino acids, both free amino acids as well as small peptides that are then taken up in the jejunum. And so it is that part of the intestines where, if one can interfere with phenylalanine metabolism, one may have a chance of lowering blood Phe levels in PKU patients. So The question then is, can we generate a PAL-like enzyme that can tolerate the stomach as well as the intestinal environment so that Phe can be removed during the digestive process? And as Dr. Harding already alluded to, our digestive tract is a very hostile environment for protein. It has evolved naturally over billions of years to degrade protein. And so this is probably going to be very difficult. And indeed, this has been, the idea has been around for a long time, since the 60s. There was a paper published in The Lancet in 1980 by Hoskins and co-workers who, for the first time, took a PAL-like enzyme. They protected it with some gelatin-type capsule. They administered it to a single PKU patient, and they saw a 25% reduction in blood Phe levels. That work was followed by a lot of attempts from various scientists to protect the enzyme from a relatively harsh environment. And BioMarin and some academic groups that they worked with, they did a lot of work in that area as well. None of that really provided an enzyme that could be administered orally to lower blood Phe levels. So others tried, yet we decided to give it a try. And the reason that we decided to give it a try is that when we started this program in 2012, we had already 15 years of experience in making very stable and productive enzymes. And we historically make enzymes that are used for small molecule pharmaceutical manufacturing. So if you know people who are on atorvastatin, that is a small molecule drug that is manufactured using enzymes that Codexis has engineered. Similarly, if you are from Europe and you use simvastatin, another cholesterol-lowering drug that is manufactured using an enzyme that Codexis engineered. And probably the biggest product that we're currently involved in is Januvia, Merck's diabetes product. We make in the order of 10 metric tons of enzyme that are used to make all the Januvia in the world for diabetes patients. So we can engineer enzymes, and we are quite good at it. So personally, I got first exposed to the opportunity of developing a PAL enzyme for potential treatment of PKU in 1999. And for a variety of reasons, business reasons, that just never was picked up as a project until Codexis was in a pretty difficult time back in 2012, late 2012, and we decided to give it a try. So that's when we started the project, and here we are. So enzyme engineering, what does that mean? Engineering typically involves finding a very complex solution, or finding a solution to a very complex problem. And it's not something that is easily identifiable in the lab or just by doing computer simulations. And to give you just a broad perspective on engineering and solutions and finding solutions and so on, I want to take you back to the Rubik's Cube. The biggest selling toy in the world. Everybody here probably knows it and had one, or still has one. And as you know, Rubik's Cube has 6 colors. There's 26 positions, or 26 outside positions on the Rubik's Cube. And the total number of potential ways that you can manipulate the Rubik's Cube is in the order of 10 to the 19th. So that is 1 billion times 1 billion, also known as a quintillion. And you know, for any of us to try all these permutations in our lifetime, we would be able to do about 1 billion a second. So that's not going to happen, right? So this is a complex problem, but the human brain has developed strategies to actually figure this out. And so currently the world record of solving the Rubik's Cube stands at a whopping 4 seconds, which is quite amazing. So put that in perspective of enzyme engineering. And so this is a picture of the PAL enzyme. It's composed of 4 identical subunits. And as you know, protein is composed of 20 amino acids. So rather than 6 colors in the Rubik's Cube, we deal with 20 colors. And very often for a optimized enzyme, we need to change in the order of 25 different positions within the enzyme. And in the PAL enzyme, there are 550 different positions. So we need to come up with the right ways of improving this enzyme. And overall, there's 3 times 10 to the 100th different ways of doing that. Obviously, we don't have that much time. So the way our technology solves this is by doing iterative work in the labs. It's a lot of high-throughput work, high-throughput molecular biology, high-throughput screening, a lot of bioinformatics to decipher all the data that we get. And once we have all the data, then we repeat that process with the better mutations that we made. So over the course of this program, we screened 27,000 different PAL enzymes. And to put that in a bit more context, so we started with about 5,000. We picked the best 10 from that. We made new libraries. We screened more, picked the best one, started making new libraries, screened more, and so on. And we went through that process 9 times. And that gave us CDX6114. You see a prototype of the drug here. It's in a capsule format, and that was used in some of our animal studies. And so the properties of 6114 are as follows. The enzyme is highly tolerant to the digestive enzymes in the intestines. It's highly stable to the acidic environment in the stomach. We actually made an effort to reduce the number of phenylalanine amino acids in the protein. So any natural protein comes with phenylalanine in it. We made an effort to remove those because we didn't want to introduce additional phenylalanine. We also engineered the enzyme to be easy to manufacture. And with the 3 products that I mentioned earlier, we have a tremendous experience in enzyme manufacturing. And this enzyme falls right into our wheelhouse. And we engineered it such that it is more stable and has a longer half-life at room temperature. So we improved all those things, but of course, at the same time, we needed to retain its normal activity. And that is what we accomplished. So in the next 2 slides, I want to talk to you about some of the in vitro and in vivo work we did. Initially, we did work in the PKU mouse, as you would expect, and we saw in the order of 30% Phe reduction with a single dose. But very quickly, we learned that we needed to go to larger animals, and that's what we did. So first, just looking at the in vitro data. So 6114 is more stable to the acidity in the stomach, and this is illustrated in the left side panel. So what we do here is we take AVPAL and CDX6114, and we expose it to low pH, pH 4, for up to 6 hours. And you see CDX6114 is highly stable. It has a half-life of, 37 weeks, I believe. AVPAL is not super unstable, but it starts to fall apart. And when you think about that falling apart of the enzyme in the stomach, that basically then has subsequent issues in the intestines where it will be degraded even faster. In the panel on the right, we show here how the enzyme is stabilized towards proteolytic attack. AVPAL has a half-life of 23 minutes. Our enzyme has a half-life of 17 hours. And so, again, historically, people have tried to make this enzyme stable by encapsulating it in a variety of different polymeric molecules or by putting PEG onto the surface of the protein. What we do, and this may seem somewhat strange to this audience, we generally affiliate mutations with a negative impact. And in our field, that is actually quite different. For us, a mutation is just a change from an amino acid to a different amino acid. Very often, these amino acid changes are bad, but in some cases, they are better. And what we do through our iterative enzyme engineering, we accumulate these beneficial mutations. And they may just be slightly beneficial. Maybe they increase the stability by a factor of 2 or improve the activity by a factor of 1.5. But if you have enough of those mutations together, you can really boost the performance of the enzyme. And by making 22 mutations in the enzyme, that is what we did. And that gives this very significant increase in stability in the intestines. So as I mentioned, we took the enzyme into a number of animal studies. Started with the the PKU mouse, then we went into the rat, then we went into the dog. And the reason that we went through this progression is that we did not really understand phenylalanine metabolism in the intestines very well. And as we went through this progression, we learned more and more. The enzyme actually turned out to be better and better. And when we then finally went into non-human primates, it started to look even better than that. And on this slide, these are 3 different illustrations of the results we obtained with 6114. Maybe I'll start with the one in the middle, the time curve. And what you see here is, this is an experiment where the monkey received a solid meal, and 5 minutes after it took in the solid meal, we gave it a dose of the enzyme, for instance, in the capsule that I showed in one of the earlier slides. And what you then you can then follow the activity of the enzyme by taking blood samples from the animal at indicated time points in this graph. The green line basically indicates how much cinnamic acid is present in the blood of the monkeys. You see that spikes very quickly. Phe metabolism is extremely fast. There's an abundance of Phe in the intestines, and the effect lasts for 6 hours, maybe slightly longer than that. And the blue line is basically a vehicle control. So that gives you an indication of how we measure the efficacy of the enzyme. Of course, you can do this experiment with different amounts of enzyme administered to the animal. And so in the graph on the left, the dose-response chart, we look at the amount of cinnamic acid that is being formed at different doses of the enzyme. And you see at the very low doses, you see it increase up to the fourth point in the graph at the dose of 12 mg/kg, we reach a maximum amount of cinnamic acid formed. And this is in monkeys who received a milk as a protein dose. And after the 12 mg/kg, the response is pretty stable. And so we're very excited about this. Now, you may wonder, why do you have all those data points after 12 mg/kg? The reason is that was what was indicated by the dog study. So actually, we started at the higher doses and then had to walk back to really find a nice dose range. So we're pretty happy to see at the dose of about 12 mg/kg, we get a pretty stable result. You may wonder how repeatable is this, how reproducible is it in for instance, the same animal. And the graph on the right shows the repeat time curve. So we took the same monkey and we administered the drug 3 times in a row. And you see the result is very similar day after day after day. So that made us very comfortable with the efficacy of CDX-6114. You may wonder, well, how does this really relate then to the disease? And I tried to do that in the next slide. And so 6114 is an orally administered GI active enzyme. I should say that we have measured or we have looked for enzyme in plasma in the blood of the monkeys, and we have not been able to identify it. So we don't think the enzyme is taken up systemically and really performs its function in the GI. So when we think about the recommended amounts of phenylalanine that healthy people take in, or that the FDA recommends for intake, and how much phenylalanine is recommended for intake by PKU patients, we know there's a difference, obviously, and the difference is in the range of 18 to 28 milligrams per kilogram. So if we would be able to remove 18 to 28 milligrams per kilogram of phenylalanine, we may be able to get into that clinically relevant range. And so we know that in monkeys, a single dose removed 12 mg/kg of Phe. And if you multiply that by 2 or 3 for twice-a-day dosing or 3 times a day dosing, what one may expect that we would remove 24 to 36 mg/kg, which is right in the range of where we would want to be. So we're very hopeful that we will see a good response from this molecule in the clinic. The path to the clinic is pretty much at a high level as follows. We're starting a healthy volunteer phase 1 study in Australia later this summer. The reason that we do this in Australia is really for business reasons. It's cost and speed. And that is a study that will be run by Codexis. If all goes well, then a company that we partnered with, Nestlé Health Science, they have taken an option to take our program from us at the end of this phase 1A study. And so if they exercise that option, they will be doing studies from that point forward. And you may know Nestlé Health Science, perhaps because that's the company that actually owns VitaFlow, which is a very familiar company to everybody here. So that is the enzyme, that the oral PAL that we have developed. That is how we got here and the path forward. There's a lot of people who have been involved in this program. Like I said, it started with 2 people 5 years ago. And highlighted in green at the top here, Nick Agard, he worked on this program for the first 3 years, got so excited about drug development that he decided to go to Genentech, a big biotech organization in the Bay Area, and is developing many drugs there now. Chimping Ching and Casey Hallows and Nikki Dallas, they completed the project, the ultimate generation of 6114. Kristen Skvorek, known to many of you here, the second ever NPKUA fellow. We ran into each other at the PKU Alliance meeting 2 years ago, and 3 months later she was with Codexis, thankfully. So she has really helped us with the advancement of the animal studies and ultimately getting the package completely together. On the far right, Rob Wilson, he's in charge of manufacturing, and we have manufactured this enzyme under GMP capabilities, of course, for the clinical study that we're starting soon. And at the bottom, our collaborators for the initial PKU studies. We ran those at the University of Groningen in the Netherlands. Christine Sarkeesian helped us with the design of those studies, and that gave us the first thumbs up that this was going to be a good candidate to advance. So with that, I'd like to thank all of you for your attention, and I'm happy to take any questions. Thank you.
Hi, great work. Quick question. Did you see active enzyme in the monkey stool?
In the monkey serum?
Stool.
In the monkey stool. The answer to the question is no, we have not seen it in the stool. We haven't looked for it either in the monkey. We did look at it in the stool of the dogs, and in some of the animals we saw less than 0.1% of PAL activity was still available in the stool. But that was in some of the animals at much higher doses.
So the 12 milligram per kilogram drop in the monkeys, what was the control on that? Like if they had not gotten the enzyme, was there any drop?
I'm sorry. So the sound is very bad up here. So you were asking for the blood feed drop?
The 12 milligram per kilogram drop that you saw, what was the control, like in the monkeys that did not get it, was there any drop?
Okay, I see. So what we measure and what we showed in the graph was the amount of cinnamic acid that is formed. In the control animals, there is no cinnamic acid formed. There is no cinnamic acid in the diet, and monkeys or people, we normally don't make cinnamic acid. So the amount was 0. What I did not show here is that when monkeys eat, when they eat a solid meal or when we give them a gavage of milk, you see a Phe spike, and that basically goes up for about an hour, and then it comes back down. That spike is actually greatly reduced, and it basically stays flat in the monkeys that receive CDX-6114.
Thank you. Dr. Nichols?
So this morning, I will present to you work from my clinical colleagues at the University of Pittsburgh, particularly Dr. Ira Fox, Jerry Vockley, Kyle Saltis, and Rakesh Sindhi. Myself, I'm not actually involved in this work, so I'll hopefully get details across to you. So this is work involved in hepatocyte transplantation of a currently single patient with PKU, and my colleagues now have a NIH grant to perform a small clinical trial of additional patients. So the first patient was a 27-year-old female with PKU who had 2 previously identified mutations in PAH, classical PKU. Her diet was not managed at all, so she had uncontrolled Phe levels of around 1,500 micromolar, and a series of clinical features of uncontrolled, because of the uncontrolled diet. And so the transplantation process had 3 stages. There's an initial stage of radiation therapy to a small part of the right lobe of the liver. The transplantation is then performed by a catheter through the portal vein, and the hepatocytes are injected through this process into the right part of the liver. The transfusion occurred 3 times over a 24-hour period. Every 6 to 8 hours, there was a injection, and a total of 5 billion hepatocytes were transplanted. This was actually about half the planned dose. During that 24 hours, the donor hepatocytes underwent a decrease in viability, and so they were not able to transfer as many as desired. There was a preset threshold for how viable the cells would be, needed to be. And then the third stage of the clinical procedure after transplantation is immunosuppression, for which the treatment regime went through several different stages over the first few months and then up to a year. And so this just shows the catheter and the hepatocytes being transplanted into the umbilical vein. There was some contrast injected to show where the cells were going into the liver. And then post transplantation, studies were done with biopsies at 3 months and 6 months, and the donor cells in this case were from a male individual. A patient, as I mentioned, was female, and so you're able to look at fluorescence in situ hybridization with probes for the X chromosome and the Y chromosome, and in red are the probe for the X chromosome, and in green are the probe for the Y chromosome. So you can see there are cells that are present in the liver biopsy from the donor hepatocytes. And this is immunostaining for PAH enzyme, and you can see a cluster in about the center of the slide. There's a cluster of brown cells representing the staining pattern for cells that are positive for active PAH. And so you can see there's just a small cluster of positive cells in this particular biopsy. And looking at Phe levels, before transplantation, as I mentioned, the Phe levels were around 1,500 micromolar, and they fell at multiple test points to around 850 micromolar. So it wasn't wasn't to the therapeutic range, but it was a significant drop of 35 to 40% in Phe levels. This was during this post-transplantation period, the patient maintained a uncontrolled diet. And so for the planned clinical trial, the inclusion criteria include that the individuals need to be 14 years or older. They have to have a previous diagnosis of classical PKU with known mutations. They need to have had poor control of their diet, standard therapy based on at least 2 consecutive Phe levels. This is around, I think it's around 600 to 800 micromolar. Baseline IQ of greater than 70. There's a few exclusion criteria, which is if the patient is known to have an allergy to immune suppression medications, if they have other illnesses or diseases that would indicate that transplant should not be done, if they're pregnant or breastfeeding, or if they're HIV positive. And if anyone is interested in learning more about the study, they can contact one of these 2 study coordinators, Angela Shinsky or Sandra Braden, and I can leave this contact information up here for a few moments. That's everything. If there are any questions, I'll do my best to answer, but as I say, I'm not a clinician and this is not my work, so if needed, we can contact the study coordinators.